So now we're going to move on to the cell cycle. So we talked about this in class a little bit, and this is basically going to be the life cycle of a cell. So in this picture here, you can see that it's going to be composed of G1, S, G2, M, and then C. So G1, let's just say we've got a cell right here and it is split into two. So this is a nice new cell, right? So what's going to happen is that cell is going to go into G1, and G1 is just the primary growth phase for the cell. So it's just growing. The next one you have is the S phase, and S stands for synthesis. And so what's going to happen here is right at this point where these meet is when the cell is going to decide whether or not it is going to divide or not. Now there's some cells that'll stay in G1 the whole time. So like your nerve cells are going to do that because they don't like to regenerate. Anything past G1 is preparing for mitosis. So you've got the G1, primary growth, then you've got S. That stands for DNA synthesis, which means the DNA has been copied. After that phase, you're going to go into G2, and in the G2 phase is going to be a little bit more growth and then copying of the organelles. And then you're going to have mitosis, so we're going to go through those phases in a second, followed by cytokinesis, which is the actual splitting of the cell. So that's going to be the cell cycle. Some of them can go through that in an hour or two, others never get all the way through it, so it totally depends on the type of cell we're talking about. Okay, so now we're going to get into the phases of mitosis, and the first one is going to be interphase. Interphase is just basically preparing. Inter in Latin roots of words means between. So this is just between mitoses, whatever the plural of that is. So what's going to happen is in interphase, all of the DNA is going to remain uncoiled. It's not going to be coiled into chromosomes. It is going to coil into those chromosomes right at the end of interphase if it gets a signal that it's going to start going through all the crazy phases of mitosis. So what else is going to happen is you're going to have your, that's a close-up, but let's look here. So it's not shown in this picture, but on either side of these sister chromatids, you're going to have something called a kinetochore. And the kinetochore is going to be this thing right here, and it's a protein. And what's going to happen is microtubules are going to reach out and attach to this. And that way what they can do is push and pull the chromosomes around, kind of like a little puppet show. Okay, so let's go through the phases of mitosis. So these are pretty basic, and the good news is in chapter 13 when we go over meiosis, a lot of these are going to be very similar. So if we look at the picture, right, well that's not, no, okay. All right, these are the phases of mitosis right here. So prophase is going to be where the chromosomes are actually going to condense, and the nuclear envelope is going to start to break down. So that's why it's kind of drawn with a dotted line there. The next phase is metaphase. Meta means middle. So if you look, all the chromosomes are lined up down the middle. Now the one thing I want you to remember is that this purple one here is an exact duplicate of this purple one here. This red one here is an exact duplicate of this one here. So in the next phase, anaphase, you can see that they're getting split apart, but these are all identical copies of one another, which means when the cell splits, you're going to have identical cells. That's really important. So in anaphase, what's going to happen is the sister chromatids, which are together here, are going to get split apart. And that's going to happen using those microtubules attached to those kinetochores we just talked about. Then the last phase is telophase. And in telophase, what's going to happen is the nuclear envelopes are going to start to reform, and the cell is just going to start to pinch a little bit in the middle. And that's basically going to be the phases of mitosis. Now, if we go a little further down, you can actually see what they look like in a plant cell. So prophase, you can see that the chromatin is starting to condense. You still see your nuclear envelope. The end of prophase, you can start to see the chromosomes, but they're still within that envelope. Metaphase, they're all lined up down the middle. Anaphase, you can see that they're pulling apart from one another. And then telophase, you can already start to see something called a cell plate, which is where the cell is actually going to split into two. And they're kind of balling up and relaxing a little bit because that's when the nuclear envelope is reforming. Now, here's another version of it. So you've got prophase, Then you have metaphase, where they're all lined up, anaphase, where they kind of start to pull apart, telophase, where they're almost apart, and then cytokinesis would be where they actually do break apart. Now, the cytokinesis, where they actually do break apart, is going to be different depending on what type of cells we're talking about. So here is an animal cell. And the way you can tell is because what's happening is the cell is actually splitting from the outside in. So this little ring of microfilaments is going to kind of cinch around it and pinch it into two. That's going to be very indicative of an animal cell splitting. A plant cell, when it splits, is going to have a harder time because don't forget it has that cell wall. So what's going to happen when you have a plant cell splitting is you can see here you've got these little bubbles that are going to form in the middle called the cell plate. Eventually they're going to fuse together and make a true cell plate, and that's going to grow from the inside out to the walls and eventually split that way. So that has to happen because that cinching process can't happen on a plant cell because it has a cell wall. Now the third way I don't have a picture of, but what's going to happen in the third way is going to happen in protists and fungi. So protists are going to be things like an amoeba. Fungi is going to be stuff like mushrooms. And what they're going to do is all of those phases of mitosis is going to occur completely inside the nucleus. Then after mitosis is complete, the nucleus is going to divide, and then the cells divide. So that's how that's going to happen. So in the next one, we're going to talk about the cell cycle and the checkpoints that we have. And that's a pretty intense one, so I want to make sure that you can go back and look at that one specifically over and over again.